So what we're going to do right now before we move forward learning more about DNA transcription is just play with a simulation. Not a big deal at all. I want to make a note, and you make a note of this, that this simulation is using the term gene expression to talk about DNA transcription. So the expression of a gene into a protein is another term for the transcription of the gene into a code that can then be translated into a protein. So gene expression is really getting at that same idea as DNA transcription. So here's how we're going to go about doing it. You have here three different simulations to play around with. One called expression, one called mRNA. Okay, that's going to be stand for messenger RNA, and then here a multiple cell one. I'm going to show you how to use the expression one, and then I'm going to show you how to use the messenger RNA one, and I'm going to just let you play around here with the multiple cell one. After you get done playing with all of them, I'm going to ask you some questions about what you think each of the things do. Okay, so it's sort of an open sandbox right now, and here's how you do it. Once you click on this one expression, down here at the bottom, that's how you're going to navigate between the other simulations. Okay, so here we are on expression. We're inside the nucleus, and we have a long strand of DNA, and we know from that central dogma that DNA codes for RNA, and then RNA codes for protein. Okay, so they're showing the DNA is this double helix here, and they're showing these proteins here. You have a diamond protein, a hexagonal protein, and a cylindrical protein. Remember, structure, molecular structure, relates to function, and your job is just to collect these proteins. And when you've collected your proteins from this gene, this segment of DNA, you're going to move on to the next gene until you finish the simulation. So what do? Well, up here we have a bunch of tools or proteins in and of themselves that can actually cause this process of DNA transcription and then ultimate translation into a protein to occur. We have something called a positive transcription factor. We have these things called RNA polymerases that you already talked about. We have these things called ribosomes. We have messenger RNA destroyers, and these things called negative transcription factors. So what you're going to do is just click on them. So let's start with the positive transcription factor. Click on it, and if I stay clicked on it, notice it shows me exactly where to put it. So I'm going to put that right there. Okay. I'm going to click now on an RNA polymerase. Oh, and if I hold on to it, it shows me where to put it. Right there. And there it goes. Okay. And what's coming off is this messenger RNA that's neatly folded messenger RNA. So what I'm going to click on now is this ribosome. And it tells me to put it there. And when the ribosome goes up next to the messenger RNA, oh, a diamond protein comes out. So it's telling me to put it right there. So I've collected a protein. Okay. And now, oh, it's putting another one out there. Awesome. Okay, so now what I have here is an mRNA destroyer. I'm going to click on that, oh, and it tells me to put it right there, and it looks like it's going to crunch up all that messenger RNA, which is no longer needed because we've coded for the necessary proteins from the messenger RNA. We have this thing called a negative transcription factor. If I put it right there, oh, it doesn't do anything right now. So what I want you to do is play with this. Do it in the order I did. Try and do these things in a different order. And when you're done, you're going to move on to the next gene. Okay, so keep going until you've completed all of the genes here. When you're done, you're going to go down here to this messenger RNA region. And what this is going to do is it's going to allow you now, remember we had these positive transcription factors. It turned out that these do something. And what we're going to do is mess with the concentration of these positive transcription factors and their affinity or how well they bind to that gene or that region of DNA right before the gene actually. And then you're going to mess with the affinity of the RNA polymerase in general. So here I'm going to add increase the concentration of that transcription factor. Okay, I might decrease its affinity here. Okay, and there the RNA polymerase binded. I'm going to remove the transcription factors entirely. and then I'm going to decrease the affinity there, increase the affinity here, so you can play around with these. Okay. Now here, I'm going to let you go ahead and play with this on your own to see how it works. Okay, so have fun with that.